Hello, good morning, everyone. My name is Sandhya Sharma. Today, I will be holding your lecture as Dr. Wasim Asgar is busy with some commitments. Today, I will be holding the entire lecture, and in my presentation, I will be talking about my research project, which I'm currently working on. And in the presentation is going to be for about an hour, and you will have later some time to ask me questions. And I would suggest you to write down your question on a piece of paper so that you do not forget in towards the end of the presentation. So I'm going to go ahead and start with the presentation. So first, talking about me, my name is Sandhya Sharma. I'm a PhD student and a research assistant in Dr. Wasim Asgar's lab. In our lab, we mainly work with the micro and nanotechnology in medicine to solve the real-world problem and develop a diagnostic test for infectious diseases, circulating tumor cell detection, and several other biological materials. I did my undergrad, my major was biotechnology, and in my master's, I took major bioengineering from Florida Atlantic University. And currently, I'm doing my PhD in electrical engineering, and my main area of research is nanobiotechnology. So these are my publications so far. I recently published, last year I published two research paper, and in 2018 I published circulating tumor cell review paper. And now I'm going to get started with the presentation. So talking about background of my projects, a sudden eruption of infectious diseases has caused catastrophic impact, causing many deaths and leaving lifelong several health issues in many others. In the past 200 years, several contagious diseases have spread worldwide, killing more than 90 million people. HIV, hepatitis C virus, influenza virus, four types of coronavirus, Zika, Ebola are such few infectious diseases that have already impacted the human life. These infectious diseases grab world attention from time to time in developing and developed nations. The effective way to control the spread of infectious diseases is to quickly identify these diseases and stop the spread, and that helps with the help of healthcare system. As you can see, here is an image of a spread of infectious diseases worldwide from the year 1998 to 2018. As you can see, there are a lot of diseases which have been spread during this time, during the recent years. So, for the detection of these infectious diseases, sophisticated lab locations are required and expensive equipment. These are some limitation which is faced by the people who are living in a poor resource setting areas. However, in the recent times, point of care diagnostic testing has become a powerful source to improve the existing healthcare system and speed up the disease diagnosis. Point of care testings are user friendly, low cost, quick, and simple, which do not require complex workroom and can work with a few resources. So here in our lab, we work mainly to develop these microfluidic platform which can work at a point of care settings for the detection of the infectious diseases. So these are during my PhD, I have been involved in several projects. Total of 4 projects, out of which 3 of them they are related to the infectious diseases, and I recently published a paper on Zika virus. Currently I'm working with SARS-CoV-2, COVID-19, and hepatitis C virus. In addition to the infectious disease detection, I'm also working on the project which is a sperm cell sorting on a small microfluidic platform. So as I have mentioned, first, talking about the infectious disease project, as I previously mentioned, the current diagnostic methods have some limitations. However, still they are being opted by most of the places. The gold standard for the disease diagnosis is RT-PCR. However, it is very expensive. The turnaround time is 2 to 3 days. In addition to that, a trained personnel is required to process the sample. So therefore, RT-PCR cannot be used everywhere. Second is a serological testing, which are antibody-based testing. These are quick and they produce results within 5 minutes. The time is like from 5 to 30 minutes. However, the ELISA or these antibody-based tests have some limitation that same type of antibody is produced by different diseases. So therefore, it provides the cross-reactivity. So therefore, we cannot rely on these antibody-based tests. So in order to overcome the limitation of the current existing methods, we came up with an approach where we can detect the virus at a very low cost and rapidly. So talking about my first project, it has been published in Archives of Pathology and Laboratory Medicine, Lab on a Chip Zika Detection for RT-LAMP based assay for point of care settings. A little bit background about Zika. Zika virus is a RNA flavivirus, which is mainly transmitted by mosquitoes. However, the other transmission mode could be blood transfusion, pregnancy. If a mother is has a confirmed Zika, then the fetus can get it. Sexual contact. So the neurological deficit is one of the impact that has been left by the Zika disease. Guillain-Barré syndrome in adults, where the person is left paralyzed, and congenital microcephaly for in the newborns, where you can see the head of the baby is smaller than the normal healthy baby. Talking about the sign and the symptom of the Zika virus are redness in the eyes, fever, headache, rashes, muscle pain, or joint pain. These symptoms can be easily confused with the other abroviruses such as dengue, chikungunya, Nile West fever, as these all viruses belong to a same family. These all are abroviruses. The structure, not only the family, their structure and the symptoms are so similar that the doctor get confused if a person is having a dengue or a Zika virus. However, Zika virus leave much more adverse effect in a human than the dengue and the chikungunya. So therefore, in order to distinguish Zika from the other flaviviruses, a specific Zika specific detection method is required that can distinguish the Zika from the other flaviviruses. So this is the image this is the layout of the approach that we have designed. Here, the developed microfluidic chip, which is made up of PMMA, contains 4 different chambers which have their specific function. Chamber 1 is used for the lysing and the binding. This is based on a magnetic bead isolation method. In the first chamber, we introduce the saliva sample or the blood plasma sample in the chamber 1, and the magnetic bead present in the chamber. Once the virus is lysed, they will carry the DNA or RNA on its surface, and the chip is placed on a platform which has a small magnet situated underneath, and under the influence of the magnetic field, the magnetic particles will start moving from chamber 1 towards chamber 4. Chamber 2 and the chamber 3 will contain 2 different types of washing buffers that will wash the beads, ensuring only the target reaches the chamber 4. Once the magnetic bead reaches the chamber 4 is already maintained at an isothermal temperature of 70 degrees. The beads will elute the DNA and the RNA from the human and from the virus into the chamber 4. And the chamber 4 will contain master mix of leucocrystal violet dye for the naked eye detection and the Zika specific primer. If the Zika target is present in the chamber 4, the primers will bind to the target and will start amplifying. And once there is a lot of amount of DNA in the chamber 4, the color of the chamber will convert from colorless or almost light blue to dark violet color. So this is the overall approach of the projects which I am working on. So first, talking about the loop-mediated isothermal amplification. Loop-mediated isothermal amplification is a process in which the DNA or a RNA is amplified at an isothermal temperature, and in the presence of 4 to 6 primers, the target is amplified. Talking briefly about the process, as you can see, there are like multiple primers which are already present in the solution, and first primer will go and bind to the target, and it will start elongating, and then in the next step, here F3 primer would go and bind to another site, and that will displace the first elongated strand. And this process will happen from both ends of the target, and eventually forming a loop-like structure, like as we can see. So this process happened at an isothermal at an isothermal temperature. Therefore, it is a faster process than an RT-PCR. However, in RT-PCR, 3 different temperatures are required to amplify a target DNA or RNA. So first, how we designed our targets and the primer. So I opted like 5 North and the South American strain from NCBI. I downloaded the sequences from the NCBI database and then aligned those 50 strains in a multiple sequence alignment tool, which is called Clustal Omega. Once the alignment was done, multiple conserved sites were manually checked, and after all those multiple sites, I picked out a conserved sequence which was 429 base pair longs. This conserved sequence was further blasted in the NCBI so that in order to make sure that this is homologous to Zika and this is very different from the other abroviruses and the other diseases. Once it was ensured that the conserved sequence is homologous to Zika, then it was further subjected to an icon software to design the primer for the loop-mediated isothermal amplification. Once the primers were designed, they were further blasted against the other abroviruses, ensuring that these primers are specific to Zika, not to any other abrovirus or any other infectious diseases. So in order to validate the preliminary testing, the target was propagated in form of DNA. So once the target and the primer were received from IDT, the different 1:10 serial dilution was prepared, and multiple different reactions were carried out with a different concentration of the target to see the sensitivity and the specificity of the primers that we designed. So in the very first testing, as you can see, this is the amplification plot of the benchtop assay with a different concentration of Zika virus per reaction. One reaction of the loop-mediated isothermal amplification contains master mix, primer, water, target, and the dye for the detection of the amplification signals. So initially, all the preliminary tests, they were carried out in a thermocycler in the PR. So as you can see, these are the preliminary tests which ensure that the primers, they exclusively bind only to the target, and the amplification is seen in the reaction where the Zika target is present. The sensitivity we got in the preliminary testing was 10 to the 100 copies per reaction. In addition to the sensitivity, we also checked the specificity, and for the specificity, we carried out in one of our reaction, we inserted high amount of HIV target, and as we can see from the plot, no amplification was observed in the HIV reaction, validating that these primers are specific to Zika. However, one of the limitations working in the lab is that using too many primer causes primer dimer. That means 2 primers within the reaction, they bind themselves and they start to amplify, which provides a false positive result. So here, as you can see, the PM reaction, which is a primer master mix reaction, does not show any sign of amplification, validating that the primers, they exclusively bind only to the target, they do not bind within themselves, and hence amplifying, hence avoiding the false positive results. So these results were further validated on a 1% gel electrophoresis, and long sharp bands are seen in the well where the amplification occurred, and no sharp bands could be seen where no amplification happened. So we further plotted the baseline subtracted amplification points for the different concentration of the target, and we can see higher the concentration of the target, lower is the time for the amplification. And we observed a good fitted straight line with a R square value of 0.885. One thing I would like to mention in here, in the process of lamp, the curve will not always be exponential, because as I have shown you in the previous slides, that once the targets start amplifying, the longer segments are produced. Simultaneously with the longer segments, the smaller segments are always formed. So therefore, it is not necessary that the R square value is always we'll get a good value of R square value. It could vary anywhere from 0.8 to 0.95. So that is one of our reasons we observe a lower R square value of 0.885. So further, in order to make the test user-friendly, and we tested a naked eye dye, which is leucocrystal violet dye. In the presence of double-stranded DNA, the leucocrystal violet dye changes its color from colorless to the dark violet color, as you can see here. 4 to 5 microliter of dye is inserted in each reaction, and then the reaction was subjected to an isothermal temperature in the PCR. So after 30 minutes, we can see the amplification occurred in the reaction which contained the different concentration of target. However, we did not see amplification. We did not see color change, which implies that no amplification occurred in the reaction carrying only 10 copies of Zika target, HIV, and primer master mix reaction. So the sensitivity with the dye remains the same as without the dye, without the colorimetry dye. So we further plotted the gray value with the help of an image chase software, and we observed a trend that the reaction which did not show color change has a higher intensity value than 175. This is a user defined threshold value, and the reaction which show the change in the color has a lower intensity value than 175. So first, after the sensitivity and the specificity test, we further spiked the propagated target into the human plasma samples with a different concentration from 10 to the power 2 to 10 to the power 7 copies per mil. This range is relevant to the actual range of the target present in the patient suffering from Zika. So we propagated the plasma sample with different concentration, and we used Dynabead-Selene viral RNA kit to extract the target from the blood plasma sample. Once the target was eluted in the elution buffer, we used that elution buffer and carried out the LAMP reaction. The LAMP reaction again contained master mix, fluorescent dye to detect the fluorescent signal, Zika specific primer, target, and water. And as we can see, the amplification is observed in the reaction containing different concentration of target And again, to validate the specificity, we carried out one HIV test, and no amplification is seen with the HIV in the HIV reaction. At the same time, no primer dimer formation in the primer master mix reaction is observed. These results were further validated with a 1% of gel electrophoresis, and long sharp bands could be seen in the well containing different concentration of target products. Again, we plotted the baseline value of the targets, and we can see different concentration showed different amplification time, and we observe a good fitted line with the R-square value of 0.936. Further, the previous preliminary tests were done based on the propagated DNA. In order to ensure that the kit we are using for the isolation of the target from the plasma sample and the master mix primer, if they work with the real sample or not, we obtained 2 cultured and 2 plasma Zika sample and 1 dengue sample from the lab, and we processed them with the help of the same kit. Once all the target was eluted in the elution buffer, the elution buffer was further used to carry the lamp reaction. And as you can see from the fluorescent plot here, that the reaction containing different Zika sample showed the sign of amplification. At the same time, dengue sample did not show any sign of amplification, validating that these primers are specific to Zika virus, and does not provide false positive results with any other abroviruses such as dengue in this case. And further, we validated these test results with the gel electrophoresis and observed long sharp band in the well containing Zika targets. So this is the overall image of the project. In the first one, in the A, you can see the overall illustration of the benchtop nucleic acid extraction from the plasma with the viral kit. These are the process which happens at the benchtop. We designed all these, and we compiled all of them in a small microfluidic chip, which is probably a size of a palm. This microfluidic chip is made up of PMMA, contain different chamber which carry all these benchtop process on one single platform. And the image on the right, C, is a color representation of the chip filled with the different type of buffer and the reagent. Here, chamber 1 will contain the target plasma and the magnetic beads. Chamber 2, chamber 3 will contain 2 different type of washing buffer, and chamber 4 will contain master mix Zika specific primer for the detection of the target if present in the plasma. And these elliptical valving chamber contains the 15 CST oil in them. The oil is filled between these chamber in order to keep the 2 buffers separate, and also the easy movement of the bead from one chamber to another. And this is the automated microfluidic platform we have designed, we have in our lab. A is an image of an Arduino, which controls a magnetic platform, means movement of the magnetic field from magnetic beads from one chamber to another. And on the right, the C is an image of the heater, which is attached on top of the chamber 4 in order to maintain an isothermal temperature of 70 degrees. So this is a small quick video of the project that will help you understanding the process better. This is the time-lapse video how the beads are being washed in the multiple chambers, and then this is the elution of the target. And once the target is eluted, the chamber 4 is further incubated for 30 minutes, and color change is observed if the Zika is present, and no color change is observed if the Zika target is not present. So After this is the result of the fully automatic chip run, which I did with a different concentration of the target. Here, A is an image of the chamber 4 before the experimentation is carried out. B, C, D, E are the endpoint results of the chip when the plasma with a different concentration of Zika target was introduced to the introduction chamber, and FGH are the negative control and 10 Zika copies per mil. Talking about F and G, these are the negative control where an unbiased plasma is ran and no color change was observed. And in G is a HIV target was introduced in the plasma, and again, once we do not see color change. Further, in order to validate the sensitivity of the platform, we lowered the copy from 10 to the power 8, 10 to the power 4, 3, 2, and we further went to 10 copies per mil. And the H is an image of that chip run where the Zika copies were very low and we did not see amplification in the chamber. So this validates that with this setup, the sensitivity we obtained is 10 to the power 2 copies per mil in the plasma, and this is the lowest copy number which is present in the actual Zika patient. So again, we plotted the gray intensity value of the results, and we observed that the plasma which had no Zika target, the chamber 4 show value which is higher than 175, and the chambers and the plasma which contains Zika target, the color change was observed, and the intensity value of the chamber 4 was lower than 175. The same trend is it is the same trend as we saw off chip. So on chip and off chip, they follow the same trend of the intensity value, helps validating that our microfluidic platform is working well as in the bench as with the similar to the benchtop experiments. So then my next project is hepatitis C virus molecular diagnostic assay at a point of care settings. Talking a little bit of background of the hepatitis C virus, more than 70 million people worldwide are suffering from chronic infection of hepatitis C virus. Hepatitis C virus causes chronic liver diseases, cirrhosis, and hepatocellular carcinoma. Effort to develop a hepatitis C virus vaccination started more than 25 years ago when the virus was identified. The virus also mutates rapidly, making it difficult for an immune system to keep up. This is the reason that we do not have the vaccine for the hepatitis C virus so far. Hepatitis C virus has a 7 genotype, which can be differ from each other as much as 70%. However, the 5' UTR and the hyperenveloped E region are the highly conserved sequence in all the hepatitis C virus genomes. The current standard method for testing hepatitis C virus is RT-PCR test, and other than that is again the antibody test, which sometimes provide a cross reactivity, and the antibody test also have a low sensitivity. Sometimes that's why they miss detecting the virus in a person. Therefore, in order to overcome these limitations, we design the microfluidic platform where we can detect the virus on a single platform based on a loop-mediated isothermal amplification. Talking about the primer and the target, we opted 5' UTR region from UF1 RNA isolated strain. We blasted that region against the other abroviruses and observed that 5' UTR region is highly conserved and is not present in any other virus. We opted that target and and then we subjected that conserved sequence in a software to design the primers. Once the primers were designed, they were again blasted against the other viruses, ensuring that the primers are highly specific to hepatitis C virus, not to any other virus. Again, for the preliminary testing, we propagated the target in form of DNA. Once the target was obtained from IDT company, 1:10 serial dilution were prepared, and assay was carried out with a different concentration of target per reaction. As we can see, the amplification in the reaction containing HCV target is seen, and the sensitivity obtained from this assay is 1 copy of the target per reaction. At the same time, no primer dimer formation is observed, eliminating the odds that the primers will provide the false positive result. At the same time, the reaction carrying 0 HCV target show no sign of amplification, hence validating the specificity and the sensitivity of the primers. Next, we plotted the baseline value, and we observed a good fitted line with the R-square value of 0.967. These results were further validated on a 1.5% gel electrophoresis, and the products were stained with a bromophenol dye, and long sharp bands are seen in the well containing the amplified product with a different concentration, and no bands are observed in a primer master mix reaction, and in the well carrying the product from 0 copy of the target per reaction. So, and after these preliminary testing of the bench of the sensitivity and the specificity, we spied the human blood plasma with a different virus copy number. The HCV virus cultured sample was obtained from and the virus was spiked at a different concentration, and 100 microliter of plasma of each sample was used for the extraction of the target with the help of Dynabead-Silane viral RNA kit. Once the target was eluted in an elution buffer, it was further subjected for the RT lamp process in a thermocycler, and amplification was observed in the reaction containing different concentration of the target eluted from the plasma containing the initially containing the hepatitis C virus particles, and no amplification is observed in an unbiased plasma which do not contain any hepatitis virus particle, and at the same time no primer master mix reaction amplification was observed in the reaction. So again, we plotted the baseline value, and we observed a good fitted line with a R-square value of 0.86. And these results were further validated with 1.5% of gel electrophoresis, where the resultant products were stained with a bromophenol blue dye, and as you can see, long sharp bands are observed in a reaction containing the lamp products, and no bands are formed in the reaction containing where amplification did not occur. So further, after the preliminary testing to make it user-friendly on the platform, we validated these results with the colorimetry detection dye, and we again ran the experiment in the presence of leucocrystal violet dye, and where the amplification occur, we can see the dark violet coloration in the reaction, and no dark violet coloration is observed in the reaction where no amplification occur. So again, we plotted the intensity value, and it followed the same trend as we previously observed. The reaction showing no color change has a intensity value higher than 175, and the reaction showing color change has a intensity value lower than 175. So far I have these preliminary tests, benchtop test, and I am working on the on-chip testing, and once I have the on-chip testing, the paper will be ready for the publication. So now talking about the third project, it is a point-of-care based RNA detection of novel SARS-CoV-2 virus, screening of pandemic. So as of January 28, we have seen earlier this year there was like a rising trend. The number of SARS-CoV-2 were all-time high, and now we started to see a downward trend. And daily new deaths are all-time high currently. So talking about SARS-CoV-2. SARS-CoV-2 was first detected in 2019 in Wuhan, China, and was declared pandemic in March 2020. SARS-CoV-2 causes a severe respiratory illness in the people with the with a compromised immune system and with the pre-existing disease conditions. SARS-CoV-2 is a RNA envelope with the crown-like spikes, with the crown-like appearance, which are famously known as like spike glycoprotein. And the SARS-CoV-2 is a 30,000 nucleotide base pair long, and the diameter of the virus is anywhere between 60 to 140 nanometer. The symptom of the SARS-CoV-2 is fever, dry cough, shortness of breath. However, according to CDC, 30 to 40% of the infected people do not show any sign and symptom. This is due to their immune system, or they already have the antibody in their system that prevents them getting the virus. However, elderly, immune compromised, and the people with the pre-existing condition which have a weaker immune system have a higher chances of getting severe COVID-19 disease. So talking about the current testing method, RT-PCR remains the gold standard, and in addition to the RT-PCR, several serological tests are also been opted, which are quick and rapid. However, they lack sensitivity. Talking about the RT-PCR, it has like a higher turnaround time, which is 2 to 3 days. At the same time, the test itself is expensive. Under the emergency use authorization, FDA has approved several other tests. However, one or the other limitation also comes with these tests in terms of sensitivity or specificity. Back in November, we had some good news regarding vaccine. mRNA-based vaccine were produced by Pfizer BioNTech and how it works. Talking a little bit about it, mRNA molecule is introduced in a lipid bilayer membrane into the human into the human cells, and that mRNA encodes for the spike protein. That spike protein further produces the antibodies that will inhibit the binding of the actual virus to the human ACE2 receptor. Current limitation to this vaccine is the refrigeration is minus 70 degrees. Not all the places they have a minus 70 degrees refrigerator, so that is currently it's challenging. However, a few questions still remain unanswered, which are like how if the vaccine will prevent the transmission and the spread, asymptomatic spread, and also no data has been there with that how long the immunity will last with the vaccine. The next is the Moderna vaccine, which is another mRNA vaccine, and it is as same as the Pfizer vaccine, and it has been like approved by FDA, and currently the data published from this vaccine has shown non-human primate results, which were published in New England Journal of Medicine, has shown that it neutralizes the activity of the virus and also limit the inflammation, thereby preventing the viral replication in the lungs after it has been administered. So as of like we have some good results regarding these vaccines. So talking about the testing method, this is an image which compares the current testing method and the testing method which we have developed. let's say if a person is having any symptom, fever, fatigue, dry cough, sore throat, or shortness of breath, the person currently goes to the testing center, and nasal swab of the person is collected, which is uneasy and it is quite painful. After that, the test results come in 2 to 4 days. The limitation to the current testing method is it is expensive. The turnaround time is 2 to 4 days, and at the same time, trained personnel is required for the processing of the sample. The one limitation when the trained personnel is involved, it could also lead to a false positive result if in case the personnel it mix up the results with some other samples. So thereby we can get a false positive result. So on the left hand side is a comparison of our current method and the method which we have developed. It is very easy. We just collect the saliva sample in a sterile tube, and then a small amount of saliva is introduced to the chamber 1, and then the automated platform will take care of the sample processing and amplification, and thereby providing the naked eye results. The positive point with this testing is the turnaround time is 60 minutes. It is low cost and no trained personnel is required as after the sample is inserted in the chamber 1. So talking about the primers and the target, how I designed it, I downloaded 75 COVID-19 sequences from NCBI, and then again aligned them with the sequence alignment tool Cluster Omega, and observed the multiple highly conserved target sequence which are conserved among the humans. Then that conserved sequence was further blasted against the SARS which occurred in 2003 in order to validate that the target is exclusively only for the current COVID-19. Once it was validated that that the conserved sequence is conserved with the SARS-CoV-2, it was further blasted against the other viruses. And then the primers were designed. Once the primers were designed, they were further blasted against the other viruses, ensuring that these primers are highly specific to SARS-CoV-2, not to any other viruses. So in the initial testing, we obtained positive and the negative RNA, positive control and the negative control as RNA sample of the SARS-CoV-2, and the negative control was SARS from 2003, and we ran the RT lamp with a different concentration of a target, and as you can see from the results, violet coloration is observed in the reaction containing positive control, and no amplification is observed, implies with the colorless tubes showing that showing that the primers are exclusively binding only to the SARS-CoV-2 virus target, not to any other virus. At the same time, no coloration was observed in the primer master mix reaction, validating that primers, they do not bind to themselves, and eliminating the odds of false positive results. So again, we plotted the gray intensity value, where intensity value was measured using image A, and again the reactions which do not show color change has an intensity value higher than 175, and the reactions which show the color change has the intensity value lower than 175. So as of throughout the 3 projects, we have seen the trend that the intensity value of these reactions, they follow the same trend in terms of intensity value. So further, these results were validated by 1.5% gel electrophoresis, and long sharp bands are observed in the well containing the lamp amplification product, and no sharp bands are observed in the negative control and the primer master mix reaction. Again, after these preliminary sensitivity and specificity tests, we further spiked the inactivated SARS-CoV-2 virus in a saliva sample. The saliva sample was further processed with a dynabethylene viral RNA kit where the target was isolated and eluted in elution buffer. Once the target was eluted in an elution buffer, it was further subjected to RT lamp in a thermocycler with a different concentration, and as you can see, the color change was observed in a reaction containing different concentration of the target extracted from the saliva, and no color change was observed in prima master mix reaction. And the negative control in this, the negative control was an unbiased plasma, which was not spiked with any sort of virus. At the same time, we observe a sensitivity of up to 3.5 times 10 to the power 3 copies per mil reaction. According to a few studies recently published, the RNA level in the saliva ranges from 10 to the power 4 to the 10 to the power 8 copies per mil. So therefore, from these studies, we have obtained the lowest limit. We went further the lowest limit, and we can validate that this specific testing would be able to detect the lowest copies of the target of the SARS-CoV-2 target from any saliva sample. So again, we further plotted the intensity value, and we observed that the reaction which did not show color change has an intensity value higher than 175, and the reactions which showed the color change has an intensity value lower than 175. Again, these results were further validated by 1% gel electrophoresis, where the long sharp bands were seen in the reactions in the wells carrying the amplicons of SARS-CoV-2, and no bands are seen in the wells where no reaction occurred. So I am done with my off-chip testing. These are the preliminary benchtop testing, and now I'm currently working on the on-chip testing, where different saliva samples spiked with the different virus particles are introduced on chip and processed on the developed microfluidic platform. So the fourth project I'm working on is selection of healthy sperm based on positive rheotaxis using microfluidic chip. Talking a little bit background, according to clinical reports, 10% of the couple worldwide are infertile, and almost half of these infertility cases are due to male infertility factor. It could be due to this low sperm count, low sperm motility, or higher percentage of the DNA fragmentation in the sperm cell. Assisted reproductive technologies have made it possible for these couples to achieve fertility. However, these infertility treatments are expensive, and at the same time, the initial step for these procedure is selection of sperm cell based on the morphology, motility, and a good DNA fragmentation index. So the current method which are used for the sperm isolation the first one is the density gradient centrifugation, where the sperm cell are placed on the top and then they are centrifuged for some time, and then the motile sperm, they settle down at the bottom. The second one is a swim up method, where the semen sample is introduced at the bottom of a test tube, and the sperm the media is placed, HTF is placed on the top, and the tube is left slantingly for like about an hour, and then the healthy sperm cell, they will swim up, and the healthy and the motile sperm cells are collected from the top. However, these current conventional methods come with some limitations. First off, they are time consuming. The process takes about 3 to 4 hours, and at the same time they are labor intense. Collection of the sample from the test tube depends upon the person to person. If the person is trained, he or she will be able to collect a good sample from the top, and if not, then he might mess up and collect the unhealthy sperm cell from the bottom. Second, the the conventional method, they involve centrifugation, which induces higher DNA fragmentation because of the higher shear stress which are being experienced by the sperm cells, which also damages the sperm morphology. In addition to that, they also produces high level of reductive oxidation species, which is not healthy for the sperm cell. And if a sperm cell is not in a good shape, it will fail to fertilize the egg cell. So, talking about the female genital tract and the guiding mechanism, once the sperm cell are introduced in the female genital tract, in the lower genital tract, a lot of sperm cells, they started moving towards the egg cell. And the first, from the lower genital tract, they started to swim upwards. Why they are swimming upward? That is because of the phenomena which is known as rheotaxis. Rheotaxis is a phenomena where the sperm cell, they flow against the flow which has been discharged by the female genital tract. Once they reach to the fallopian tube, two other mechanisms, which is thermotaxis and the chemotaxis, further guide these cells to infuse to the egg cell. However, the major determinant of this distance determinant for these cells is rheotaxis. So by taking the advantage of this natural phenomena, we design a microfluidic chip in which we isolate the sperm cell based on a rheotaxis phenomena. So this is the schematic of the microfluidic chip which we have designed. This is a sample inlet chamber where a small amount of semen sample is introduced, and this is the flow input chamber where a small amount of flow is introduced, and which the flow is from collecting chamber towards the sample inlet chamber. Once a sample is introduced in the sample inlet chamber, the chip is incubated for an hour. Based on the rheotaxis phenomena, the healthy and the motile sperm cell will start swimming against the flow and will start getting collected in the collecting chamber. The unhealthy sperm cell will get washed away in the collecting chamber. Bottom right, you can see the 3D image of the microfluidic chip which we have designed. This image is obtained in a console software. So this is an actual microfluidic chip which we have designed. It is a PDMS based chip which isolate the healthy and motile sperm cell from the semen sample. We further simulated the microfluidic chip in a console to look at the velocity profile and the shear stress. And as you can see from the first image, from the image A, we can see there is a very low velocity magnitude in the collecting chamber. Therefore, the sperm cell can retain in the collecting chamber for a longer time without getting exhausted. Sometimes if they get exhausted, they again lose their motility. So that's why this microfluidic chip has like a very low velocity profile, which which provide a sperm cell a very safe and healthy environment where they can move freely and can be collected for a longer time. This is image B is an image of it's a dissectional image of a channel B where you can see a little bit higher velocity is present in the channel B, which actually helps isolating the healthy sperm cell from the non-healthy sperm cell. And then image C is a shear profile of shear stress, and where you can see the shear stress is higher on the against the wall and is a little bit lower in the center. So all these profiles actually help in filtering the healthy sperm cell from the other one, and only healthy and motile sperm cell will be able to travel towards the collecting chamber. The other will get washed away. So we examined the collected sample from the collecting chamber. We compared three different data. First one from stock, second one from the chip which contained flow, and as a control we ran another chip which contained no flow. And as we can see from the graph, the sample with flow contained all 100% motile sperm cell, and with the no flow, many dead sperm cells were observed, and the stock had contained maximum of dead cell than the alive one. So this chip were able to isolate 100% motile sperm cell. Next is the isolation efficiency. As there is a flow rate and it filters a healthy sperm cell, a fewer number of cells were obtained in the collecting chamber than with the no flow chip collecting chamber sample. Next, we simulated the velocity profile of the sperm cell and observed that with flow sample has a higher curvilinear velocity in comparison to no flow and the stock, which means the sperm cell, they are a fast traveler and will be able to move quickly and to foretell the egg cell. So next we did the morphological analysis of the cells, and for the normal sperm cell, it has to have like an oval shaped head, long tail about 45 micrometer long, and has to have no large vacuoles, and also it should be filled with 40 to 70% of the acrosome. So image 1 is you can see it's a normal healthy sperm cell, and the abnormality can be observed in image 4 and 6. Image 4 and 6, you can see it's a pyriform head, and in the number 4 the tail is kind of bent. So these are considered unhealthy, morphologically abnormal cell according to the WHO. So these are some images from the with flow where we observed a high number of morphologically normal sperm cell, and according to one of the study published in 1990, it is observed that if a morphologically healthy sperm cell is chosen for the fertilization, the chances of fertility increase from 40 to 97%. Therefore, this microfluidic chip, it provide a pool of healthy normal sperm cell which can significantly increase the fertilization ratio of the assistive reproductive technologies. So this is a control chip images where you can see majority of the sperm cell with the abnormal parameters, and this is the percentage of the normal sperm cell isolated from the collecting chamber. The with flow group has shown the highest percentage of morphologically normal sperm cell in comparison to no flow and stock. So I'm on the last part of this project where I am trying to profile the DNA fragmentation index of the sperm cell collected from the three different samples. And once the DNA profiling is done, I will be ready for my next paper to publish. Next, do you have any questions related to the project?
No, I don't have any question.
Okay.
I have a question.
Yes.
So, hi, Sonia. It's Tosh, by the way.
Hi.
When you did the chip, you put the sperm sample in and everything. Did you have something to help it flow into the collection chamber, or did you just did it just flow by itself? Like I was confused. Like what's the motive to separate them?
Okay, so as I said, the motive to separate them is like based on rheotaxis, where the sperms
Okay.
they swim against the flow. So as you can see, this is the schematic of the microfluidic chip. It contained four different chambers, right? I'm sorry, I forgot to like specify that in the thing. So the four chambers, first one is a flow input chamber where a small amount of fluid is introduced and which flow from flow input chamber towards the collecting chamber. And when the sample is introduced in the sample inlet chamber, as you can see here, the sperm cell, they will swim against the flow and will start collecting in the collecting chamber.
All right, makes sense.
Okay, yeah. All right, guys, any other question? Tosh, do you guys take attendance for the online class?
I don't know if he does, or if he just looks at who comes. I'm not aware.
Yeah. I think everything is online, so everybody can watch the lecture again or watch it if they miss it at any given time.
Okay.
And I think you can really see who watched it. I mean, that's what it used to be. I don't know if it's still like that.
I don't.
You can
I don't
see
think he
it can
does.
see who watch it live and it can see who watch it later. So if nobody watch it, he will know.
Okay. Okay. Yeah. Okay. Any other question, guys? I guess your class is up to like 10:50, right? I believe.
Mm-hmm.
So anything you guys want to discuss or like, put your input towards your projects or anything, like you're more than welcome to discuss. Because this is actually his class is nanobiotechnologies. This is we pretty much work with in our lab. And not just, yes.
Do we have access to your presentation?
I'm pretty sure like I am recording the lecture. That's what I've been told.
Okay.
So
Yeah, I know.
I don't know.
But I'm
Like,
saying, can we have the paper and print it out or something?
I'm gonna have to ask Dr. Asghar about it. For the one project, I have already published it. This paper is already out there,
Oh, I understand. Okay.
right? And the other two projects are similar because we have not published it yet. So I don't know if I can share it with you guys publicly as of now.
Okay. I know.
But this one is already out there. You can read it and you will have a better understanding regarding the other project. Like this one, Zika one, we actually like in the first project, we were actually making sure that our platform works, right? The currently which I'm working right now is based on RNA and the real samples. So initially we made our platform work. Now we are working with the actual sample and because like working with RNA is always challenging. So now I'm at that part with the hepatitis C virus and the SARS-CoV-2 virus. So guys, I guess if you guys have no question, we are good to go.
Yeah, that's fine.
Right? Yeah, in case you guys don't have any questions, so there is no point of holding you guys. Right? All right.
Okay. Thank you.
Thank you.
Thank you.
Thank you, Saya.
Bye. Bye, Tosh. Thanks.
Bye.